Alright, welcome back to the bonus TPM function class. So we're going to take all that stuff we just did and we're going to make a function from it. And this is going to be really cool. I'm going to clean my environment just so you can see that I am not using something that's already been in there. And we went through this. There's an exercise for you, but now we're going to see how to make your own function. And it's really quite simple. So we're going to call this function plot gene expression, and we're going to do all the steps we did, but then next time we want to use it, we just say plot gene expression and put in genes of interest, whatever gene you want to plot, or genes, it can be tons of genes, and then what the data frame is, that's this df here. So you can see when you write in function, R knows what you're trying to do here, and it needs to know what the parameters of that function are. It's sort of like when we've done any function within the parentheses, we have to put something in, a comma, and another thing. Those are called parameters to the function, things that the function absolutely needs to do its job. Sort of like multiplication needs two numbers to get into it. Here we're saying we need a list of genes and a data frame. Same thing we did up above. Okay, so we are now in this function here because of the curly bracket, and it's going to end down here, so you can see it was a lot of work we did to get here, and there's where the function will end. So let's just go through it briefly because you have just done this class. But so what we're going to say is pretend like we have a data frame in our head of those TPM values, and we're going to say for that data frame, grab the input gene of interest we had and index those. So we're going to take only the rows that match your genes of interest. So you can have the massive data frame of all the genes, and it's going to go in and just grab the ones you told it you're interested in plotting. And we're going to do row names to column to bring those, now that smaller data frame, bring the row names in. And we're going to take this df plot genes that we just made, which is our subset, and the variable that we're going to move from the meta into a column is the gene ID of what we just merged with. Again, if this all sounds complicated, it's really the exact same steps we did up above, just plotted or just written out. But it's hard to visualize when you're not actually having something in your environment to look at. Okay, so to plot the genes, we did this pivot longer, and we removed the gene ID, and we took names to time and replicate, and the values that were previously wide are now long, and we're going to call that new column TPM, which is going to have multiple entries for each gene. We're going to take that and do that separation thing we had just done a minute ago, where we're going to separate the name of, so this would require that your data frame has the same naming system that we did. And you're going to separate by the underscore and get rid of that wild type variable and just keep time and replicate. And can you guess what's next? Group by. So we're going to group by the gene ID and the time point, and then get an average and standard deviation. First we're going to convert time point to numeric, and then we're going to group by gene ID and summarize the mean and standard deviation for that grouping, and then plot it. Okay? So all of that is now going to be one line of code. So we're going to run the function to load it into our environment. Okay, so now we have a new function here, like same when you did library, tidyverse, or whatever. Really, we just did this, but it's our own custom function. You can go look at it by clicking here, or it tells you what the parameters are too. And then let's just give it an example of the same genes we made before. So this is just an object that's just listing these genes, which are only being used to go in and index into the bigger data frame and grab just these ones out. And now watch this. One line, so genes of interest and TPM filtered, same inputs we started with. Oh, I erased TPM filtered. Oh no. Okay, so I have to go back up and load the TPM data that we started with, because I erased my environment to show that this would work without any memory of what we had just done. Now we are at the end of this function. Okay, so now I have a TPM filtered object in here. that's the data frame I need. It's required for this function. And there we go. Voila, one line of code, and we got back this same plot. And I think this gene looks quite interesting. Okay, so that is it. So now, how do we have to run that function every single time? I don't think you're going to want to do that. So what you do is you go in our files, you go up to your utility folder, which is above lessons in your cloned master class. We made a util here, and our first function lesson is here. And I made two functions for you in here. You have cloned this, so you have it. This is just showing how a function works, like just multiplying two numbers is technically a function. What are the parameters? How do you set this up? So you want to have a description of your function, the parameters, that one's the simple one. So now here's our plot gene expression function that we just made. So you could copy and paste all that code. I even left the markdown in here. Exactly the same thing. We even did this example usage. That's nice for people to see what it's going to do. And so now if we source this R script of 00, our first function, lessons.r, we're going to have that function. And that's just like we could put up with library, what we do at the beginning. We would just use source instead of library. And so yeah, so it's telling you what the parameters are, a list of genes. TPM requires this format. That would be really important if you wanted to share this function. And as a double bonus, since we're on like the bonus round, I made another function for you called log-fold change plot. And so this does the same thing we did for TPM, but it's going to plot the log-fold change values from our DE-seq. So I highly recommend you go through and get that. And it's here a few different G subs and mutations, but you'll see that a lot of the stuff is going to be the same. And so if you really want to know more about functions, here you go. And you will have two functions, one that will make you a TPM plot for any gene, and the log fold change from 0 to 12, 24, 48, 96. And so you can compare them. But I think it's really cool that now we just do source this util here, function lessons from R, and now we have all that function information from that R script here in our util folder. Always keep them in your util because you don't want to mess up your clean thing here. And so this one needs genes of interest, which we already have in our environment. So do we? Yep, genes of interest, and a resdf. Now that came from our DE-seq object, our data that we loaded in. So let's just do this real quick. Load back in our DE-seq results so we get some form of resdf. Go all the way to the bottom with the function. We sourced it. We have those loaded, but it's making sure you actually load in ggplot. What happened here? Okay, so our genes of interest, it's not liking that. But you can go through here and it'll make you log fold change plot with your resdf. We didn't make a use case of this one. We have a resdf, so that's good, and we have a genes of interest. It doesn't like the format it's in, but if you do all of this the same, we can do that, and we sourced it already, and it still doesn't like our genes of interest. So in the function annotation, you might have to make those genes of interest into a data frame, but you can go through this and get to that. So really this is a lot of reiterations of making functions, how to source them, where to put them, and then in the future, if we wanted to use this, we would take this command here and put it up when we're loading our up here. And we're going to make that log fold change plot anyways. It's just, this is more of a bonus exercise on how to make functions and how cool it is that whenever you do a lot of code like that, like take a table, make it longer, group by, you can turn that into a function quite easily. And then you never have to do that again in the future. Okay, moving on, we are going to make heat maps in the next lesson. So see you there.